So while we are waiting for hopefully other people to show up, okay, if they don't, we can start without them. But there's this, but really quick. Oh my gosh, now I put this up here and I can't see past this. Okay, hold on really quick. I had a little possum. We found a possum in my yard. Well, my dogs found the possum and like grabbed it with their mouths. Well, Meatball did. And he like had it, you know how dogs will do like pick up a big rat or something and then like kind of like toss it back in their mouth further. Well, he did that and I yelled at him and I was like, maybe don't do that. So he dropped him and he's okay. This is him after that. He was running around the yard, but he doesn't know where to go. He's too tiny to like get, over the fences or anything. Doesn't really know what he's doing. So we called like a wildlife rescue group. They're gonna come and hopefully pick him up sometime tomorrow, I think. But yeah, he's in a little we have him in a little plastic box thing with a little bit of water. He doesn't like it at all being in there, but that's where he is for now. This is the second time. This literally happened last semester too. The two littler, even littler, if you can imagine, baby tiny possums. So I think they come from my neighbor next door. I don't live in like a rural area. I live in Bethany. So, but there's a lot of possums here, man, and a lot of raccoons. I think it's just because we're so close to Stinchcombe. But wanted to share that with you guys because it is cute. Some people think that possums are gross, but I think that they are cute. So here's our thing.
So do you think the wildlife will, like kill them, or will they actually keep them? Because they don't they multiply a lot?
Yeah. So I don't know. I mean, the lady last time this happened, they came out all the way from Hinton to like pick them up and like go and like they nurse them back to health and they just put them on their land. They have a lot of land and I don't know if they live or die after that, but it's certainly a better odds than probably throwing them back in someone else's backyard over here.
They usually rehab them and release them. If they can't be rehabbed and released, they will find people that'll put applications in to have them as pets. My clinic
So
sees all of that.
do you know if they carry germs like rabies, like a raccoon would, like in the daytime, raccoons are like rabies that seen in the day? Like how does that work with those?
Yeah, so possums can't get rabies. It's a
Oh.
weird, yeah, isn't that cool? It's sort of cool. But they are kind of like armadillos. It's rare for them to have leprosy, but like armadillos have leprosy on the outside of them. So you're not supposed to touch armadillos. They can have it on possums, but it's really unlikely in like the more urban areas for that to be the case.
Yeah, I just had to tell a guy at my clinic that possums don't carry rabies because his dog got a possum. So of course he brings his dog in for his rabies vaccines.
Oh, well, better safe than sorry, I guess. Yeah, something about, I think, like the markers on their cells, the virus can't infect them or something.
We've actually got a couple raccoons that come into the clinic too. One thinks he's a cat.
Yeah, we used to have a raccoon that would come into our sunroom, like we had a doggy door, and it would lock at night, but it would still, they're strong. So they would just push it open anyway, even though I had it locked, and they would just come in there and like eat the dog treats and like they would just sit and chill and like eat the food in the sunroom while we were sleeping at night. And so we'd come in there every morning and like there would be like another mess. We thought that we like kept it out that time, but like the lock didn't work again for whatever reason. So now we have like literally actual like weights, like would go like barbell weights, pressed up against like a metal sheet to keep the dog door closed until we can get a better solution. But we'll probably get one of those like drop down metal doors so we can close it at night because it's so much more convenient having it open in the day. I don't let them out all the time. Just wait a little bit longer and see if people want to join, and then we'll get going. So fun. Oh, it's Discord. I'm like, what is this? All right, I think we'll go ahead and get started. We've only got 4 people playing, but that's okay. We never expect too many people to join us, right, for the review, so it's okay. Some of this is going to be pretty much the same stuff. Like, there's not a whole lot more I can say about the stains, but that's okay. Review, repetition, right? Right, so it says everything we've learned so far is fair game. Like I've told you, that's not entirely true. Everything up to Kirby Bauer and not including Kirby Bauer. Kirby Bauer won't be on there. And then starting at exercise 11 with the selective and differential media, that'll be everything from there on. But the DNA stuff, the eukaryotes, Kirby Bauer, the ELISA, those will not be on there. So don't waste your time studying that. All right, so it says 26 stations. This will be true. This time we actually have 2 questions at every station except for the last 2 questions. Question 49 gets its own and question 50 gets its own. I'm gonna let you know ahead of time. Question 49 and question 50 are gonna basically be those lab report, like the worksheet like checklist or whatever, with like what you got for your results and whatever, not for yours specifically, but I'll have one filled out as like an example. And then I'll ask you to identify the gram-positive organism for 49. And then for 50, we'll have another sheet over there, and that'll be the gram-negative organism. So those get their own stations. 49 will be off in that little corner, but 50 will be up by like the TV by me. So just be aware. I'll remind you guys when we get in there, but it's a little bit different setup, but otherwise it's the same thing. It's going to be Scantron. Try to keep your Scantrons in the little cubby area. I will have everything clearly labeled as to what goes with what question and what tube is what and all of that. Almost every single tube where you have to determine a result, like is this positive or negative, you will have an uninoculated and an inoculated tube there. There will be real tubes. It's not pictures. It's tubes that were results usually from you guys if they lasted, if they kept. Some things don't keep in the fridge. So we're hoping the power came back on. I don't know if you guys know this, because most of you guys that are joining me right now are from my first lab. But right after that, in my second lab, we were almost done, but the power went out in the entire science building. Like, who's surprised, right? So who is surprised by that? The whole building went out of power. So I'm hoping that we'll have power tomorrow morning. There is a contingency plan where if it doesn't happen that way, we are going to have to postpone the lab. That's going to suck. And I don't want to have to do the lab exam. I don't want to have to do that because if we're planning on taking the test on Tuesday, I'd rather just knock it out on Tuesday. But if we have to move it to Wednesday or Thursday, I guess we can do it. But, you know, it's completely out of my control, unfortunately. So I'm going to be there at probably 8 to set everything up, hopefully. And I will let you guys know as soon as I know anything. I'm probably going to be the first one in the building. Luke or Dr. Rhodes never gets there before me. He usually either gets there at the same time or after. So I doubt he's going to be there to say anything. And sometimes he'll send me like the emails about like, oh, they switched this, the stairway or whatever. And then we get there and it's not that way. And I'm like, I can tell you haven't been into the building yet, buddy. Like, I can tell. But anyways, so I will keep you updated. We're just gonna go ahead and assume that everything's gonna be fine tomorrow. I mean, how hard can it be to fix it? So everybody just hope that it's okay. Because I really would just rather get it out of the way so you guys have the rest of the time to study for the actual lecture final. But anyways, assuming everything's okay and my cultures grew, because that's the other problem, right? Because they're in an incubator that relies on power. Because there's stuff that I have to reculture for the test. Worst case scenario, if that happened, my cultures didn't grow, but we had power anyways, I'm just gonna print out pictures, y'all. I'm just gonna print out pictures. And if I print out pictures, they're coming from that study guide that I gave you. So I'm just saying, like, and everything should be as obvious as that study guide. It's not, it shouldn't be like super, like, I can't tell, is this yellow or not? Like, it should be pretty obvious. I try to make it as obvious as possible when I pick the ones that I do pick for the exam. So 1 minute per station. Again, we'll rotate through just to keep everybody kind of cranking through each question. I feel like that worked out pretty well last time. Other than that, remember to wear your lab coats, please. Oh, I can't click, right? It's not a PowerPoint. All right, let's go over, let's start from the beginning. Culture basics. All right, what is most commonly used to inoculate cultures? The one we most commonly use. This. All right, so that is correct. It's going to be the loop. I don't know what else to say. You should know it by now. That one is easy. All right, in which quadrant of a streak plate would you expect to find the most single isolated colonies? All right, great. So we all know this one, the fourth one, and the reason is because that's where it's gonna be the most diluted out, right? So that's the idea. Yes, it's gonna be that same question on the exam, and it will be there. Good old friend, at least you guys know that one, right? All right. So yes, aseptic technique, be aware of what that is and why we do it. Lab safety, general lab safety stuff. Types of media, like that one is talking about broths versus slants versus like semi-solid versus butts and that sort of stuff, and how you would inoculate them. The basics. All right, so the stains and isolation, which the only method you need to worry about is the quad streak. Very, very widely used. All right, what is the first step in the gram stain procedure? All right, so that's correct. Most of you guys got this one. It's correct. The first step is going to be the crystal violet, and that's what's going to get stuck in that peptidoglycan layer of the gram positives. And interesting little tidbit of info. When we're talking about, and you don't need to know this for the exam, but just it correlates, okay? Just I promise it makes sense. McConkey agar. We know that McConkey agar has bile salts in it as our selective agent. It's not the only selective agent. So some of these media have more going on than what I'm teaching you about, but I just cover like basic stuff. McConkey agar also has crystal violet in it. And so you can imagine that if crystal violet is in there, that it would get stuck in the peptidoglycan layers of the gram positives and prevent them from growing that way as well. So the presence of the bile salts and the presence of the crystal violet. But the only one you're going to get tested on is the bile salts for the McConkey. So just FYI, but little tidbit makes sense when you think about how crystal violet works, right? All right. What color do properly stained gram-negative bacteria stain in the gram stain procedure? It's gram-negative. Right, so this is red. It looks pink to us, but easier to call it red. So that, purple is positive, P and P, and then red to me is negative color. So that's how I remember it. All right, what is the name of this staining procedure? right? So that's the endospore stain, pretty obvious with the red stain of the vegetative portions of the cell versus the little pill-like guys or little bubbles, right? That stain teal, kind of a teal green on the inside. So yeah. All right, which of the following bacteria will be acid-fast positive? All right, so typically when our media isn't acting up weird and I can actually get things to grow normally, we will be using one of the mycobacterium as one of our samples. I had an old sample we were able to do the acid-fast test with, but it has this sort of crumbliness, and you can tell that it's got that waxy coating compared to other bacteria. That waxy coating is very protective for them, makes them resistant. So mycobacterium, whether it's tuberculosis or leprae, which causes leprosy, or these other ones like flea or smegmatis or whatever, yeah, mycobacterium, because the mycolic acid, that waxy substance. All right, what is the name of the primary stain, it's supposed to be stain, not stain, used in the acid-fast staining procedure? It's the primary stain for that. Okay, so anytime we're talking about the primary stain for something, we're talking about typically the first stain, and that usually is going to be the most important one for the result. In this case, that's true. Carbofuscin. Carbofuscin is lipid soluble, so that waxy coating on the mycobacteria, that mycolic acid waxy coating, that carbofuscin can fuse with that because it's lipid soluble, and it won't interact with any other normal cell structures. So we will get that fuchsia color with those, so that bright pinkish, reddish color. That's very pink to me, but very bright. And then we counter stain with methylene blue, right, to give the contrast for our negative cells, which is what we saw in the image. All right, after the slide has been heat fixed, when do we use heat again in the acid fast procedure? All right, yeah, you guys got it. So we know, right? We don't use heat in that. The only one we used heat in was the endospore stain, and we used it for 10 minutes. It was moist heat to help get the malachite green inside of the endospores. Wow, that was really loud. I just got a little ding from my email, and it was just really loud in my ears. Okay, anyways, but not on acid fast, right? It just sits for 5 minutes. So what color is negative acid fast stain? I did mention this. Let's see if we remember. All right, excellent. So that's going to be blue. That's that methylene blue counter stain that we'll have for any of the cells that are negative. All right. Right, in which staining procedure do we use steam? Well, I just said it, didn't I? Alrighty, so that's going to be the endospore stain. Like I said, that's to force that malachite green into the endospore, like through its keratin-based like cortex coating. And it'll hold on to it, but it doesn't bind well to cellular structure, so it rinses out with water. And then we counter stain that one with safranin. Right, know the procedures, just like we were kind of saying, not the times that we're using them, again, same as last time, not the times, but do know what order we're using, which stains we're using, why we're using those stains, like how they work to get you the result that we're interested in, and be able to identify results from each of the stains just by looking at one slide. All right, and you guys remember from the last one, like there will be, you'll just say, what stain was this? And then there's just a slide in there. There's going to be questions like that again. And I only have a limited number of slides that I can use, like I only have a limited number, right? So most of these you will have seen before. I'm just, all right, so the new stuff. Differential and selective media. What type of media is this? This is the kind of question you're going to get on the test. And I'll just have a plate out there, hopefully with stuff growing on it. Alrighty, so this is EMB. The way that we know that is that dark color that it has, and then that metallic sheen that's going on the growth on there. I know we have a lot of the red color ones, but just try to remember that EMB is that dark, almost brownish red looking one. McConkey has more of a pink color to it. So if you can differentiate those two, then you're in a pretty good place. What is the selective agent in this media? All right, so this is correct. It is eosin methylene blue, like it says. It's literally methylene blue and eosin Y, which was the thing that was going to inhibit, related to identifying eosinophils, as well as inhibiting the gram-positive growth. But fine to say it as eosin methylene blue, if that helps us remember it, since it is EMB. It is literally what it is titled. So that one shouldn't be too difficult. It's just letters EMB. If you can remember that one has to be lined up with the words that match it. Should be okay. All right, which media is this one? Alrighty, so this one is McConkey agar. We know this because it has that pinker color to it, the pinkish red background. And then the growth that was on top of it was pink in this image, which shows that it is utilizing lactose, which might be the next question. But do remember that both McConkey and EMB will be testing for lactose use. And I'd be aware of maybe which tests might be testing for the same things as one another. All right, multi-select could be more than one. Is this media selective or differential? So it can be more than one, just FYI. All right, so it's going to be both selective and differential for this one. McConkey agar, like I was saying before, it has a selective aspect that the biosalts, which is one that we're focusing on for McConkey, that will inhibit the gram-positive growth and favor like gram-negatives. Whereas the color changing, that differential, that's the lactose usage that made the growth on the actual plate look pink as opposed to just whatever color it normally looks. That is a thing. There's a reason that it changed to that color. It's not just turning that color because the media just is that color. All right, what media is this one? Excellent, guys. So that is in fact mannitol salt agar. So we've got a selective agent being the salt, literally salt. And then we've got the differential agent being the phenol red, which you gotta love it. It's in like most of the stuff. So the phenol red, now we've become so used to it, it's probably no big deal to say that we know that it's an orangey color. We know that yellow is acid and that pink is going to be alkaline because we've seen it so many times. Hopefully for you guys, it's something you remember. All right, what is the pH indicator used in this medium? Of course, I already just said it. Until I don't really look at the questions at the time. Yeah. Phenol reds, I'll write these like a semester before. Of course, I'm using the same stuff as last semester, but I didn't know to look through it beforehand to like prep myself. So I'm really bad about giving away my answers. That's okay though. All right, so phenol red, of course, is our pH indicator for that one. So definitely know what selective means, what differential means, what the difference is between those two, that you can have both. You can have one or the other or neither, right? You can also have media that might be just enriched, like blood agar. Blood agar is differential as well, right? It tells you that if you have hemolysis going on, so it's enriched and differential. And I would be aware of the different kinds of hemolysis as well. Know what the color changes mean or any change in the media whatsoever, like when we have gelatin liquid, what did that mean? Be aware of, I guess we're gonna talk about the tubes here in a little bit, but yeah, so color changes in these media. We didn't get to see the metallic sheen on the EMB, but it can do that. I just wish I'd had a sample to make it do that, but no, we weren't lucky. So pH indicators, what they are and how they work. And also don't forget about staphylococcus media and blood agar and the 3 different kinds of hemolysis. So yeah, staph media pretty much just has salt going on and that's it for our purposes. Biochemical tests, lots of tubes and lots of colors, right? So let's go through them. It's mostly what this review is going to be about. So which test is this one? Okay, so this is citrate or citrate, however you want to say it. This is citrate. So we know this because it is green and then it turned blue when it was positive. There's only 2 tests that do that, and it's going to be citrate and malonate. So with citrate, that's the slant, and that's what we were looking at in the image, and malonate is going to be the liquid version. So that's how we know that that would be citrate. I'm probably going to ask about that in a second. All right, which of the tubes is positive? Okay. Okay, so that's going to be B. I know you have to kind of like look at like the positioning of the answers, which I don't care for much, but yes, B, tube B. It was one that had blue in it, and that meant we had a pH shift, right? Alkaline pH shift because we used citrate. Okay, there we go. There we go. Why is it positive? Well, let's see if we can correlate what I said at least to one of these. Okay, so the pH went up when I said there's an alkaline shift. Remember, acid is anything less than 7, and base is anything higher than 7. It'd be very key to understand at least that basic concept of pH for the exam as well, which is something we should know for the lecture exam anyways, just FYI. All right, so for the citrate agar, Simmons citrate, that's what it's officially called. How is that one inoculated? We use just the loop to do the slant. That's it, normal slant. How is it read? We look for a color change, whether it is green original or if it changed to blue. That's positive for the use of citrate. So the colors that are produced, we're only going to see blue, really, that is produced for this one. And what do the colors mean? It just means that we have alkaline aspect. So something that had a basic quality like ammonia or something, raised the pH, so higher pH. And so that's why we get the shift to the blue color. And the pH indicator in this one is bromothymol blue. All right, which test is this one? Okay, So this one is phenol red fermentation. The reason that we know that this is because we have a Durham tube in there. It has a nice orangey color. And yes, we could see that color, similar color with urease, but urease test does not have a Durham tube in it. It's probably gonna be the next question on here. It's freaking like, what is that upside down tube called? But phenol red fermentation does have that, and you do need to notice qualities like that. All right. What is the name of that upside down tube in the assay? Yeah, I was right. All right, the Durham tube, not too difficult. I try to call it that whenever we're talking about it in there. So that's not a new name, and it's the only name for any tube that's upside down, so not too bad. All right, this could be more than one. What are the two products of fermentation that can be detected in this assay? If you are fermenting, what two possible products could you make that this assay can detect? Okay, so you guys did great on this one. It's going to be gas and acid. So we're looking at the pH change. Yes, it can turn pink. We know that, right? But that's only from peptone breakdown into ammonia. And anybody can do that. That has nothing to do with fermentation. When you ferment, though, and you're making acid as your product, you'll see that orange shift to yellow, just like we did with the mannitol salt agar. And you'll also potentially see, if yours is a gas producer during fermentation, potentially see gas collecting in the actual Durham tube itself. So. All right, which test is this one? Okay, so this is the SIM test, the sulfur, indole, and motility test. Yes, I'm sure there's more questions coming up over it, and next I'm trying to keep my mouth shut about it. Okay. What does the I stand? I didn't succeed. What does the I stand for? Okay, so this is going to be indole. So I bet it's going to ask about what indole is, but if it doesn't, then we will talk about that, I promise. Okay, how is indole detected in the SIM media? Alrighty, so this is accurate. It's going to be the addition of Kovacs, and on the test you will have, typically for almost any question about media, you'll have the media there in front of you, even if it's not about inoculated media, then you'll at least have the media there to remind you of like, oh yeah, this is the one where we did blah, blah, blah. So I feel like that helps having that, and you can pick up and handle the tubes as well. Just don't spill them, of course. I feel like that makes it easier to remember. Okay, how do you detect sulfur, H2S, hydrogen sulfide, in SIM? All right, so that's gonna be the black precipitate. So before we move on, I do want to point out, like in that image, which we've seen like 3 times now, of the SIM tubes next to each other, like the regular one, and then the one with the red at the top, and then the one with the black precipitate, and then just regular goldish color Kovacs, right? We all know what the negative looks like for Kovacs, but that reddish in the middle is what it would look like if you were positive for indole using the Kovacs reagent. It is that pinkish red color, just so that we are aware in case it pops up on the test. But yeah, indole is a byproduct of tryptophan breakdown. So if your organism can break down tryptophan, it makes indole as a byproduct, and that's what you're testing for. Okay, so hydrogen sulfide is a byproduct of sulfur reduction. I know we just learned about nitrogen or went over nitrogen reduction today, or nitrate reduction today. So you get an idea of that's basically the breakdown of nitrate. Same thing going on here. We have a sulfate product in the media, and if the organism can break it down into hydrogen sulfide, then it will react accordingly with the iron in the media and cause it to be released to cause like a black precipitate to form. So that's how that works and what we're testing for with that. Sulfur reduction. How is motility detected in the SIM test? So the M, remember, is motility. How would you say that your organism was motile if you were doing this test? Right, most of our organisms in the lab were motile, so we saw that sort of clouding out of the media. Some of them cloud out nice, where you can see that they're definitely clouding out, and it's not just the whole media cloudy. And that's nice because then you can actually see and confirm, yeah, that's not just the media clouding up, that really is my organism spreading out from the stab line. When we do have non-motile organisms, which we had one, but, and I think we might have a couple, but just don't grow very well. They weren't growing very well because acid in the media, right? But stabbing it, it'll stay on the actual stab line, and you'll see it, and it might look a little kind of fuzzy almost, but it won't spread out. It'll stay right where that stab line is. So it looks nice and tight with the clear around it, and won't look fuzzy or hazy or cloudy. Yeah, cloudy is better word for it. So how do you inoculate SIM? SIM is a butt, right? So we're going to stab it with a needle. And that might be a question on the test. I'm going to ask you, how do you inoculate SIM media? You would just say, circle needle or whatever. Select B for needle or whatever. Interpreting the results and the changes. Be aware of what positive and negative look like for the sulfur test, for the indole test, and for the motility test. And then the addition of any reagents. We know we use Kovacs in this, right? So be aware of that. Test for the indole, which is made from tryptophan breakdown. So which test is this one? Uh-oh. Right, this is litmus milk. It's that milky purple color. It always starts out with that lavender. That's the neutral color for the litmus, which is our pH indicator, which is probably the freaking next question. But anyways, remember, for litmus, acid is going to be pink and alkaline is going to be blue. I would be aware of those. I'd be aware of like the possible colors you could see for litmus for sure. Like, no, okay, pink means acid, that's made from fermentation. White means reducing, right? We reduced the litmus. If you do see blue, it's alkaline from breaking down proteins in there. If we have digestion, it'll be cleared up, and that's from really breaking down all of the proteins in there. And then if we have coagulation, like all of those scenarios, you'll want to be able to know what's going on for each of the colors. Because I'm going to put a tube in front of you. It's only going to be one tube for the litmus, but I'm going to put a tube in front of you and ask you, basically, is this doing this, or potentially, what is it doing? So be aware. Righty. In the litmus milk, what color is associated with an increase in pH? That's so rickety. Okay, so blue. That blue shift in the litmus is the alkaline. So that means an increase in pH, a higher pH. So that's what we're seeing when it turns blue. Okay, in litmus milk, not link. In litmus milk, white. What does it mean? Okay, great. So white, if you see there's white, that means reduction. You can have white without it being coagulated. You can. We didn't see that in our class, but you can. It could just be like sitting there kind of like chill. In that case, and you almost always see, not every time, but almost always see reduction with pink at the top, right? That acid. And that's because when we have high enough acid production, that's when we see the reduction happening. So they often go hand in hand. But they are different. And yes, that acid production can be related to the coagulation as well. That's why we often see them all together. You can have individual ones. All right, so litmus milk, know that what your different colors mean. What is happening with digestion? Well, if it's if you have digestion, if you see it, it's going to be translucent. It's becoming translucent. I don't care if it's purple translucent or if it is like plasma translucent. I don't care about any of that. If it's translucent ing, if it's looking translucent, that is digestion. How is gas detected in litmus milk? We won't have any samples of that, so you don't really need to worry about it, but they can form little bubbles in the coagulation. What is the sugar in milk? So the sugar in milk is lactose. So this is a lactose fermentation test. We've already seen and talked about other ones that can test lactose, even in this review. So we have this one already. We have the carbohydrate fermentation. That can be lactose. We have a lactose for that, right? We have the McConkey agar, and we have the EMB agar. I think that's it so far we've looked at. But there's going to be at least one other one. So I would be aware of like when you have overlaps of tests like that. So we have a lot that are testing for lactose usage. If we have overlaps like that, I'd be aware of which ones are going to overlap. All right, which test is this? If you were to lift this up and look at it in the light, that like light area around that streak, that would be like almost clear. Okay, so this is starch hydrolysis. The actual media itself looks like normal nutrient agar. You would never be able to tell the difference. We know by inoculating it that it was a lot softer than normal nutrient agar. So you would know it that way, as well as by putting those drops of the iodine on there, and then it turns it that dark brown color, right? So in that one, we're looking for starch hydrolysis. Which organism pictured is positive for starch hydrolysis? Okay, so it's going to be the left organism. We have that kind of obvious visible halo around that streak. The other one had a streak that looked like it wasn't, creating that dark color. Sometimes the bacteria, when you put the starch color on it, itself, like the bacteria itself, can repel the iodine when you're adding that onto the starch media. So if that happens, that will look like the bacteria in that tiny little area, like it's not, interacting with the iodine. But don't let that fool you. You have to see the actual halo around the outside in order for it to be considered positive. But you guys did good. That was the right answer. All right, what is added to detect starch hydrolysis? Oh my God, I've said it already. Okay, so we had iodine. That is correct. That interacts with the starch itself. All right, which test is this one? I do want to point out there is a Durham tube in there. That's what that is in the middle. Okay, so this is the one that we did today. This is the nitrate test. That's what it looked like before we inoculated it, right? And we know that because it looks like normal, almost clearer media with the Durham tube upside down. So only the fermentation media and the nitrate are going to have the Durham tube. Those are the only ones. So you can tell by the color which one is which. All right, so what do reagents A and B detect? Okay, so A and B are detecting nitrite. Very good. I don't want to say too much, I feel like. Yeah, what does the zinc detect? Right, so you guys got that right away. So it's nitrate, yeah. So we know that we add A and B first. Easy enough. If it turns red, then it's positive and you're done. You know that your organism reduces nitrate into nitrite. If you add A and B and you don't color change, you don't know yet if it is positive or negative for nitrate reduction. So that's why we add zinc. That's the second part of that. If you need to, if you get no color change from A and B, you add zinc. Zinc is going to detect nitrate. It will turn red as a flag, a red flag that nitrate is still there. And so that means that your organism still has nitrate. You did not reduce nitrate because it is still there. So then if you see red with that as your red flag from zinc, it means you are negative for nitrate reduction, okay? So you have to be colorless after the zinc in order to have confirmed that you have some other form of nitrate reduction going on there, besides 2-nitrite, because nitrate can be converted to a whole lot of stuff. But yeah, so that's what we're testing that. That could potentially for other people that are doing these things to determine what their organisms were more specifically than we are, determine like if it made it from A and B, that tells them something. Then if it made it, had to be tested with the zinc, right? So they can use that information from there. But anyways, the zinc you add later, zinc late nitrate, right? Alrighty, know what is detected at each step. So we know nitrite and then nitrate. Red is not always positive. It's positive in the first step and negative in the next step for nitrate reduction. And then don't forget the gas. You guys, definitely the second class, you guys didn't see the gas. Kristen had gas in her gram-negative sample. And if you didn't get to see, that's okay. It looked very similar to the gas that we have forming in the carbohydrate fermentation. Actual air inside of the Durham tube itself. If you have gas present in your nitrate media, you don't need to do the test, really. Hate to break it to you, but you're automatically going to know that you must be reducing nitrate, because where did that gas come from? It's not carbon dioxide. It is nitrogen gas or a nitrogen-based gas of some kind. So it will collect that in the Durham tube, and that will tell you that you have nitrate reduction going on right away. If that's all you want to know is whether you have it or not, then that can tell you. Of course, we have learned not all nitrate reduction produces gas, right? Most of you guys didn't. And most of you guys had nitrate reduction. So yeah, just be aware of that. There is a possibility for gas collection. That's why we have the Durham tube in there. So which test is this one? Oh yeah. Okay, so this is TSIA. And we know that because it was that orange. Probably going to ask us about the phenol red, but I'm going to say it anyways. Phenol red again is our pH indicator. We have all these different possibilities for the different results that we could have. So I'm sure we're going to go over some of those results. One thing I'll say about TSIA, because there is like an opportunity for multiple kinds of results that you can get, right? Same as like with SIM, it's literally 3 tests like spelled out for you. But those have multiple tests going on. You're going to have more questions about those than any other test on the exam, than any other, of like the tube tests on the exam, just so that we're aware. I mean, that makes sense. I want to be sure that you understand each of the steps. So you will have more than one question about different kinds of results from TSIA and SIM. All right, multiple choice here. Which tube or tubes shows evidence of hydrogen sulfide production? All right, 3 and 5 is the correct answer. That's the black precipitate. It's the same as the SIM or the sulfur, same thing. That's the iron part of the TSIA, triple sugar iron assay. The iron is testing for that sulfur reduction. All right, so here's another one. Which tube or tubes show or shows evidence of gas production other than H2S? I feel like this one's obvious. Yeah, I feel like that was really obvious. But yeah, that's what it normally really does look like whenever you get appropriate gas production with your fermentation. If we could have had more organisms to work with, it would have been nice. We could have seen more of that variation. We couldn't get them to grow. That's life. So don't you love it? But yeah, that will actually lift up the media like that. And then you'll also have bubbles like on the side as well. And you can see that too with the ones that do have H2S, the black precipitate, but no, like pretty much no matter what, if you have black precipitate, that hydrogen sulfide is a gas. So you can form bubbles just from that and not even from fermentation. And so it's hard to tell the difference from that test, right? So that's why I ask that for the difference, right? So TSIA, that was that orange slant. How is it inoculated? This one was our unique one. This was the stab and the slant squiggle, right? So we're inoculating with the slant smear and stabbing with the needle. All right, how is it red? We're going to be looking at color changes, whether that is from phenol red turning yellow or phenol red turning pink or not changing. We're going to be looking at the black precipitate or the lack thereof. And we're going to be looking at gas bubble presence or breaking in the cracks in the media from the gas.
Call your daughter.
All right. I feel like I'm not going to work this. Okay. Anyways, the colors that are produced, I kind of just already said it, but yellow.
That was
or
not
black,
cool.
Or orange, or go. Or orange before that is better. Yellow, black, orange, pink. Those are pretty much the colors that are going to get produced. Feel like that's everything, yes. And then what do those colors mean? Yellow is for acid, just like it always is with phenol red. If we have no change, then that means no change. And then we have pink, that's alkaline. And if we have black, that's going to be hydrogen sulfide. So hydrogen sulfide and the presence of visible bubbles or lifting of the media or whatever, that's obvious, right? We can see that that's not an issue. Even just a little bit of bubbles in there, by the way, counts as gas. So just be aware. But what does it actually mean? Like, what does that mean if it's yellow? We know that yellow is acid from fermentation, but this is triple sugar iron agar. So triple sugar, what we're testing for is fermentation of glucose and or lactose and or sucrose. Okay, we can't tell the difference between fermentation of lactose or sucrose in this media. But we can tell the difference from those two from the glucose. Glucose will give you a yellow butt, and sucrose and lactose will give you a yellow slant. You aren't going to have sucrose and lactose without the glucose. Okay, so we don't see that typically. So, but yeah, no matter what that combination is, though, even if I were to ask you if it was yellow on the slant and pink in the butt, then you should be able to tell me, oh, then that was just lactose and sucrose being fermented, and it's pink in the butt because peptone's being digested once again into ammonia. Same kind of test going on here as what we have in our carbohydrate fermentation tests. It's the same. It's just in a solid form. It's still detecting gas as well as acid production from our sugar. And then we can even tell two kind of categories of sugars going on at the same time. But this one also adds in that iron aspect to test for the sulfur reduction. Again, our pH indicator in this one is phenol red. All right, which test is this one? Oh, yeah. Yes, this is urease, very classic example of it. Yeah, so we got that peachy orangey color of media, no Durham tube. So that automatically tells you that it's urease. And then that pink, that bright pink. Hell yeah. That's what we're used to seeing. All right, so which test is this one? Okay, so this is catalase. Do keep in mind, don't get confused between catalase and oxidase. Do what you need to do to remind yourself that hydrogen peroxide adding into, I'm just probably going to ask you that as the next question, isn't it? If you add hydrogen peroxide and to look for bubbles or not, that's the catalase test. Do what you need to do because you're catalyzing a reaction. In the oxidase test, we're looking for that cytochrome C oxidase in the electron transport chain of aerobes, which is probably also the question after that or something. All right, which test is this one? Oh, yeah. All right, so this is the oxidase test. So we already know now, because I already said, from that one tube that we had cultured, our TSA tube, you could get two tests out of it. Yeah, we started off with our oxidase with the drops on the little Q-tip and looked for that purple color, like I just showed. And then we added the hydrogen peroxide for the catalase test. And you don't need to know, like, as far as like reagents that we add to media, you need to know what they are, as far as like we use, ferric chloride for phenylalanine deaminase, or we use Kovacs reagent for SIM. But oxidase, that reagent from the dropper, I don't need you to know anything beyond oxidase reagent, okay? You don't need to know anything beyond that. And same with like Kovacs. You need to know it's Kovacs reagent and that we're testing for indole, but I don't need you to tell me what's in Kovacs. If we didn't talk about that in the class, then don't worry about that. All right, so other tests that you need to be familiar with that we didn't cover here: malonate, deaminase, MRVP, and gelatin. Malonate is our green broth that turns blue if it's positive, bright blue, right? It's a broth though. So that's the difference between citrate. It has bromothymol blue, and it is alkaline byproduct because we utilized malonate as our carbon source and made an alkaline byproduct. Deaminase, that is phenylalanine deaminase. I just didn't want to write it all out. That one, we had to add those kind of greasy yellow, they look greasy to me. I don't know if it does to anybody else, but the yellow drops that we would put in, and then you wait for the slant to turn green. And it usually happens within a couple minutes. And that whole slant should have a green tint to it when it is positive. If it's negative, you just see that yellow collect in the bottom of the tube and nothing happens. That's testing for literally the deamination or the removal of an amine group or the removal of an NH2 group from phenylalanine, which is one of the amino acids. All right, MRVP, methyl red Voges proscourer. So we're here testing for heterolactic or mixed acid fermentation. So I'm probably just going to refer to it just as mixed acid fermentation. It's different. Most of the time we're just going to make lactic acid. but these tests are going to tell you if you're making other acids besides just that. So methyl red, we add the methyl red drops. If it's orange, it's negative. It's bright red, it's like a pinkish reddish, sort of like the indole test, right? When we added the Cobax, if it's positive, same kind of color. That red is positive, right? Not orange. The VP is Vokes Pro Scour. That one was the one we had to wait 30 minutes. So we added the 2 reagents, which aren't important. I don't care what they are. Add the 2 reagents. And then you wait 30 minutes. And what was the positive for that one? Red again. So they're both red. We're both testing for different acid production than just lactose. All right, and then gelatin. It was a butt at first. Luckily, that was a nice cool day in the lab. So it actually was staying like a butt, but you stab it, right? Nice stab for that one. And it's going to be turning to liquid and staying liquid, even if I put it back in the fridge after being in the incubator. So bring it out after still being cold. Jello, most of the time, when you put it in the fridge, it's going to re-solidify. And that's what's driving that. So if your organism breaks down gelatin, which makes jello, then it will no longer have that structure. So it goes liquid for positive. That's what I'm saying. All right, basics for bacteria stuff, the stuff that I always forget to re-add on to the thing. How would you classify this organism's characteristic arrangement? I have an arrow pointing at the one that I'm more interested in. Right, and if you were looking at this and you're trying to write down your characteristic arrangements of your organism, you could put down strepto and diplo, because you see both going on in there. But yes, I was more interested in you guys being able to tell me strepto, that's the chains. All right, what is the name and shape of these bacteria? Oh, I said name and shape. Shape, what's the name of the shape, I guess? So this is bacillus. So the bacteria themselves, the cells themselves, their cell shape is bacillus. There maybe was a little bit of palisade going on that might be accurate. But here we're just looking at the shape of the cells. And I promise that on the exam, the question will be clearer than that. But okay, so bacterial cell shapes, you're only going to be able to see the bacilli or the cocci. I do have coccobacilli, but that would be mean. So let's not do that. So let's just do cocci or bacilli. That's ones you need to be able to distinguish from. Okay, so they're really tiny and you need to be able to show you're focusing on the shapes. The arrangements, you're only going to be seeing either staphylo or strepto. Okay. And don't get those confused. So don't get these two confused. Specifically, if I ask about the shape of the cell, don't give it an arrangement as your selection. And if I ask for the arrangements, don't give it a shape. That's the answer. Do remember that simple stains are a thing. So that is something that if you stain something with methylene blue, it's going to be blue. All right, microscopes, how you use them, their parts. All right, this is the easy one. Observing a 100x objective lens through a 10x ocular lens. What's the total magnification? Exactly, so it's going to be multiplied together. So that's 1,000. We've seen this one before. It's just repeating. All right, which lens is used with oil immersion in our lab? All right, so this is going to be 100x. That's exactly right. In our lab, we don't have 500x lenses. There are microscopes with that, and they probably do need immersion, but we are talking about our lab. So other things you want to know, the parts of the microscope, like objective lens. So I'm going to ask you a part of a microscope. We did objective lens on the previous exam, so it's probably not going to be objective lens on this next one. I don't know yet what it's going to be because I haven't actually gotten that far on setting it up, but I will ask you that same question, like what is this part and what does it do? Okay, it just won't be the same one as last time. Parts of the microscope, lab safety, just basic stuff like wearing a lab coat, more or less when I'm saying like lab safety, I want to remind you guys to wear a lab coat, like please wear a lab coat, okay? All of the stains, we already kind of talked about those, all the tests, all the media. You are going to need to know all that information that we are going over, and I will test you on various aspects of those on different kinds of media. I might not ask you about, you know, I don't know, like pH color change for one thing, but I might ask it on another thing, but I might ask about gas on that one and not on another, you know what I mean? So be prepared for any question on any type of media, know how they're inoculated, know the color changes, what their pH indicators are, if they have one, and that sort of stuff. All right, and on the plates, specifically on the plates, of course, because that was the selective and differential lab. So you should definitely know which ones are selective and or differential and or enriched with a growth factor for fastidious organisms, right? We know that that is the case for the blood auger. So just review that stuff, and this is very flashcard friendly information. Read an identification chart and use a dichotomous key, like I already told you about. It's going to be filling out like whether there's positive, negative on whatever tests, the whole thing will be filled out. And then I'm going to ask you, based on it had a positive for this test, a positive for this test, a positive for that test, and a negative for that test. What is the gram positive organism? And then you will look through it and then say, oh, this is gram positive. Okay, this is Staphylococcus aureus or whatever it is. So be prepared for that. That part shouldn't be too bad as long as you can like follow along in the key. So that will be on there. It will be it's the old key. I'm not just let you know that it's the old key, but it doesn't matter. I mean, I'm not asking you to memorize the key. I'm giving you the key. I'm giving you the key just so you know. You don't have to memorize that. Okay, so that is it for the lab review. Good luck. I know you don't need it. You guys are doing great. You're doing way better than my traditional classes. So you should really be proud of that, honestly, because I'm giving you the same stuff and you're doing way better than they are. Especially my online class, they always do the worst. But summer people really apply themselves. All right, so good job, guys. Any questions? Sorry, I just saw this. All right, so for what you guys need to study, and I did post this on the announcement. If you guys didn't see it, it's okay, but it is there. I will test you over labs 1 through 7, and then 11 through 17. But the stuff in that middle part, no. So you still need to know the stains and all that. Yeah. I know that some of that stuff, like everybody's like, yeah, oh yeah, we already know that. Like that's fine. It's not a thing, but yes, I do want to remind you that technically, yeah, it's going to be 1 through 7 and then 11 through 17. Any other questions? All right, I would appreciate it just because you guys are here. If you haven't already submitted your in-lab worksheet on Top Hat, I'm trying to get everybody's grades in for that as soon as possible because it's just easier for me to do that now. It's okay if you want to wait till midnight. I'm not that, angry over it for getting your in-lab worksheets done, but it's just nicer to know that that stuff is already taken care of for everybody. So if you could get that in, that'd be great. If you can't, I understand. You got to focus on studying. Just do what you can to get those done. And then remember that we do have extra credit. This has like been a mystery extra credit. It does exist, I promise, and we've been doing it for a reason. So for those of you who have done any extra credit at all, that will show up when you are done with your final exam, your lecture final, not your lab, but your lecture final. So it will show up with that. You should know what you're getting from your extra credit at this point, because you can do the math for, what each one should be worth and all that. We aren't, I guess, not getting, like, you guys didn't get course evaluation links, right? Like, they didn't tell me anything about having you guys do your evals or anything like that. And that was a big part of my extra credit. Was like 1%, and it's only 3% that you can get, but I guess a third of the extra credit. So I'll probably do something where like, it's gonna be easy enough that people can do it and most people can get the extra credit if they wanna do it. I'll let you know what that is, but that'll be sometime, I'll let you know about that on Wednesday or Thursday. Maybe even Tuesday, or yeah, maybe even Wednesday. Yeah, Tuesday, tomorrow's Tuesday, maybe even Tuesday after the exams, so we'll see. But I want to give you guys something still. My plan is I usually give people all of that extra credit just for completing the evals, but because I do read those. Those are, if you guys don't know, those are not like letting the university know how terrible your professor is. It's letting your professor know how to improve the course. So yes, the university tracks it, but like they don't actually read it. So that's how I get my information from you guys on what you want changed for the course. You know, sometimes when people are really mad and they don't like how I ran the course and they fail the course and whatever, for whatever reason, they like to put stuff on the eval saying that this professor was terrible and didn't teach anything and whatever. And they think that that's gonna like get me fired or something. That's not how that works. Just so you know, like I'm the one that reads that, not other people. So I'm just seeing you being mean if you write stuff like that. So I'd rather you be, not that you would do that, you guys are wonderful. But I'd rather you be honest. And I mean that if you do get evals for this course or any course in the future, whoever your professor is, be honest with it and tell them how you felt about the course so that they can improve the course. I don't know if other professors listen to it, but I do. And I try to change my course accordingly as I go to make it better as much as I can. So if you get it for this class, please be honest, let me know what you would want changed. If you don't get it for this class, at least do the same for future professors. So that's my thing. So, but yeah, okay. Spiel over. Anybody have any questions for lab stuff? All right. So if that's it, if you guys don't have any questions, I am going to go ahead and log off and upload this for people that come join us so they can watch it later. And I will see you tomorrow. Happy studying, and I'm sure you'll do great. No reason to think you wouldn't.